So what I want to talk to you guys today, and Natalie will be the co-presenter, will be getting to the nitty-gritty details and the really exciting data to discuss. Is NGS and applying NGS and certain pitfalls you may be facing. The one question I like to ask at the start of my presentations, and it's a question for you to consider as we go through the different slides, because what we continually ask is, what is the impact of assay failure in your laboratory, and how do you monitor for it? And this question really embodies the passion and drive at Horizon that we do want to answer this, and that there's lots of different answers that can happen and can impact your laboratory. And by developing different types of reference standards, we hope to be able to help you and support you to answer that question. Now, with next generation sequencing, it has a number of attractive qualities, and one of them, or one that resonates well with people like yourself, is you can take your tumor sample, you can extract the DNA, you put it through, you do your line prep, put it through your NGS workflow, and it then allows you to test for the status of specific genes, whether that's AKT1, BRAF, EGFR, KRAS, NRAS, and many more. And so switching from a qPCR-based method or a sequencing-based method, you can start to really extract a lot of information from just one sample. And where certainly in molecular diagnostic assays, where this could be going is in the cell-free DNA space, where you may have very limited material from blood to run such assays, or likewise if you're doing fine needle aspiration, the sample, the quantity of the sample is going to be very precious to you, and so being able to apply a workflow that can give you lots of information all in one go is a very attractive proposition. Now, what does any new technology bring? Well, new technology is there to bring you a lot of good things, and the challenge of any new technology is the variability and understanding the full workflow, so you can mitigate any problems that can arise at each of the steps. As this is a nice little workflow. You've got your tumor sample, you'll extract your DNA, you then need to quantify your DNA, it goes through a library preparation step, sequence it, and then there's a bioinformatics where you analyze an action for it. What we have developed at Horizon, and this isn't what we can talk about in today's webinar, but there are other webinars that you can listen to, is we've developed a full range of reference materials that can help you to answer questions about the variability of each of these steps. And while we are looking to develop maybe one universal reference standard, we currently have a nice panel that answers the questions at each stage. And before I hand over to Natalie, I wanted to try and get some data into my first part of the talk. And this is really nice data from AmpliSeq panel using we're using our most popular reference material. That's our quantitative multiplex. And what we did was we sent this quantitative multiplex to 3 different partners and asked them to run this and look for the specific variants. So there are 11 different oncospecific mutations at a given allelic frequency. So with all of the blue bars here, that's Horizon. So that's us. That's what we defined the reference standard to be, and then qualified that using digital PCR. And what's nice is the majority of the partners can identify all of the mutations when it's above 5%. I've put a couple of sort of highlighted rings in where actually there's some unusual results from our partners. So the EGFR deletion, which is the 3rd mutation along, 2 of the partners just couldn't identify that, and that's supposed to be around 2%. The EGFR T790M, which is 2 more along, again, a couple of the partners struggled to identify that in the sample with the AmpliSeq panel, and so on and so on and so on. And what is the key take-home message, or I guess the building block for the rest of the talk is, while next-generation sequencing opens up a number of opportunities, and it is a very exciting technology, it's critical that we all understand where the limitations may be, and what we're keen to do and very passionate about doing is providing samples that will allow you to really tease apart how good the workflow is to help identify any variability that could creep in. Before I hand over to Natalie, we are always very open to questions, and so if you did want to ask any questions during the talk, there's a little panel, I think, on your right-hand side to type away the questions, and we'll be looking to answer all of them at the end of the session. It is my great pleasure to introduce Natalie, who is our US Customer and Technical Support Scientist. Natalie has a great background in next-generation sequencing, has a PhD in chemistry from Washington University. Without carrying on talking too much, because I do enjoy doing that a lot, I shall pass you over to Natalie and enjoy Natalie's slides. Thank you.
Thank you, Jonathan. So I wanted to start by just providing a brief introduction to next-generation sequencing. And when we think about the advancement of next-generation sequencing, you can consider how the invaluable resource that is provided to researchers in multiple industries and across multiple disciplines. And it's really going to be a major driver during the personalized medicine revolution that we're in the middle of. And however, when we look at the cost of sequencing that is plotted here from the National Institutes of Health, it's not taking into consideration, that decrease in cost is not taking into consideration the significant costs associated with the infrastructure and the expertise that's required to develop a robust and routine NGS pipeline. And so if we consider what was predicted by Sponer in 2011, the cost of the sequencing portion of the experiment continues to decrease, but the cost associated with the upfront experimental design, the downstream analysis, that's now what's dominating the cost of each assay. And this is true whether you're performing a preclinical R&D project, but even more so when we're talking about clinical assays. And so in this paper, the authors note that the unpredictable and considerable human time that's spent on the upstream design and downstream analysis is really the important part of this. And as Jonathan mentioned, here at Horizon, what we're doing is we're aiming to develop tools that help researchers and clinicians optimize their workflow so that NGS can be more reliable and ultimately more affordable, because you've streamlined those resource-intensive areas. And so depending on the different genetic material that you're starting from and the experimental workflow that you follow, you can apply NGS to many applications, as I've sort of summarized here. But for today's webinar, we're focusing specifically on DNA resequencing, which implies that the data generated is going to be compared to an existing reference sequence, such as the human genome, and we're aiming to identify onco-relevant mutations, including single nucleotide variants, insertions and deletions, copy number variants, and translocations. And as Jonathan showed earlier, when we think about that full NGS workflow for resequencing applications, there's many molecular manipulations that occur in order to generate that fully analyzed data set. And in each of these steps, you can have bias and error introduced. But today we're just going to focus on the variability and the sources of error in 3 of those points. The first being the biological sample itself. The second, the molecular steps of preparing a sequencing library. And then finally, we'll dive into the specific bias associated with some of the commonly used sequencing platforms. And I just want to mention, while we won't go into detail today, if you're interested in learning more about the bioinformatics analysis, we have a recording of our previous webinar on our website, and you can access that at your leisure. And so a great reference that I found that I'll kind of go back to along the way today is this fantastic perspective article from Nature Reviews Genetics. It's titled the Role of Replicates for Error Mitigation in Next-Gen Sequencing. And so I'll kind of refer back to this workflow that they've presented along the way of the webinar. And so again, as Jonathan mentioned, in each laboratory, as a researcher or a clinician, you should really consider the impact that bias and error have on your own assay. And so as was requested in our previous webinar, I will highlight a few points throughout the webinar where known reference standards that we offer here at Horizon have been shown to be really vital in better understanding and combating this to ensure that your assays are accurate and reproducible. And these points are particularly important, again, when we're considering patient samples of low quantity, low quality, and especially when you're trying to detect low allelic frequency variants. And really, in order for NGS technology to evolve and to allow us to better discern things like haplotype phasing, determine differences between two dysfunctional gene copies, we have to have a good understanding of the error rates and the limitations of the pipelines that we're using. And so first, as I mentioned, we're going to focus on the sources of error associated with the biological sample itself. Some of these errors, such as mislabeling or sample mix-ups, are due to human error, and they may be difficult to predict. And so while you have sometimes some unknown unknowns, if you will, running a known predictable standard alongside your own samples can at least help to introduce a checkpoint in your data in that case. And so then you also have just inherent biological variability. So for example, tumor heterogeneity or mixed cell populations, and that can add additional variability to your sample. And so in this case, the use of biological replicates can help identify true positive variants in a cell population. And so we'll discuss this in a bit more detail later. And so when we're thinking about those samples where limited sample is available, this will require us to amplify the DNA by PCR in order to achieve sufficient material for sequencing. And this degree of amplification that's required will depend on the library preparation approach that you select. And then finally, you have the degree of fragmentation, whether that's inherent to the sample, such as in cfDNA, or if it's a result of the preservation method that's been used, such as in FFPE samples. So when we think about the offerings that Horizon has for FFPE or formalin compromised materials, we're really considering how these perform differently in an NGS assay with regard to variability in primer binding, amplification, or even exclusion during size selection as a result of the high fragmentation. And so what we have are these genetically equivalent multiplex reference standards, which are offered in varying formats from our HD 701, which is a high molecular weight, or with a high DNA integrity number, to formalin compromised and fragmented DNA with low DNA integrity numbers. And so these allow researchers to evaluate how fragmentation affects detection. And so specifically on this slide, I focused on HDC 749, which is a mild formalin treatment. So that has been treated with formalin, but it still is a high molecular weight and high DNA integrity number. And then HDC 751, which has been subjected to a harsh formalin treatment. And so you'll see that the DIN numbers for those are much lower. And so this quantitative multiplex range, which includes this formalin compromised DNA format, can be used as a set together to help you investigate the effect of formalin on your workflow, starting from the very beginning, from the quantification all the way through the library preparation stage. And these standards have this characterized fragmentation patterns that you can see on the slide, and they've been quantified using the qubit assay, which prevents overestimation and has been generated, is been demonstrated to happen with the nanodrop assay. And so we've targeted defined allelic frequencies, and with the comparison between the mildly treated sample and the severely treated sample, you can investigate the effect of formalin on the allelic frequency determination of your assay. So when we look at something like amplification, we compare HDC 749 and HDC 751. And again, these standards are derived from genetically equivalent cell lines. Really, the only variability we're changing is the intensity of the formalin treatment. And so you can see in the plot here, collected during our droplet digital PCR validations, the CT values for these two standards are different, both in their quantitative value, but also in their reproducibility. And this doesn't necessarily mean that HDC 751 is performing poorly, but rather it's an important reference for better understanding what expectations one should have for their own biological samples that have been treated with formalin. And importantly, the impact of this variability in the amplification will be dependent on the library approach that you're using. And this is further exemplified when we consider the allelic frequencies that we observe during our digital droplet PCR validations. And so when we compare these three matched lots of the HDC 749 and the HDC 751, where batch 1 for each is derived from an identical cell line mixture, we can observe quantitatively here the effect of the intensity of formalin treatment on our variability. So here we observe that for all lots of HDC 749, the detected allelic frequencies fit within the acceptable range that we use for normal non-formalin treated samples. However, in their matched lots where the actual frequencies are biologically identical, but the cells have been subjected to a more intense formalin treatment, some of the variants fall outside our benchmark for non-formalin treated samples. And it's interesting that not all of these are at low allelic frequencies, but rather are likely dependent on the genomic context of that region. And so again, I want to emphasize that these results don't indicate that HDC 751 is not performing properly, but rather that HDC 751 provides us with these realistic expectations and allows us to better understand the variability that we will observe in our own formalin-treated samples. And so, as I mentioned earlier, the impact of your sample variability is really affected by the approach that you take in preparing your sample for sequencing. And so now we're going to dive a little bit deeper into that library preparation step. And so while our knowledge of the human genome has been significantly enhanced since the completion of the Human Genome Project, there's still many regions that are either not well understood or very repetitive. And so as a result, it's often preferred to interrogate a reduced representation of the genome, focusing specifically on the exonic regions or the smaller targeted regions of interest with short 100 to 300 base pair sequencing reads. And given the high throughput nature of NGS, you can introduce multiple samples in a single sequencing run, which makes each assay more affordable and more efficient. And so if we think about the primary ways of approaching targeted sequencing, we'll consider solution-based enrichment, which uses probes to capture specific regions, and secondly, targeted PCR to generate short amplicons. And so reference standards can play an integral role during your protocol optimization, especially when focusing on sequencing library preparation. And it's ideal to have a standard that mimics the concentration, the genomic diversity, and the fragmentation level of your actual clinical sample. You want to ensure that there are no competing regions for probe or primer binding, that the input quantity, the quality, the concentration yields the optimal library output. And if we, again, consider the formalin compromised reference standards, we can compare whether it would be beneficial to use a PCR-free library, for example, to eliminate amplification bias, or really just to better understand how the formalin treatment affects that amplicon generation step. And as we've discussed, Horizon's reference standards are all derived from genomic DNA from cell lines, which makes them much more realistic than, say, a plasmid or a synthetic standard. And so again, parameters that you may want to evaluate at the library stage of your assay optimization would include how your library protocol performs in providing adequate sequencing coverage over your region of interest, whether your PCR or amplification steps are introducing unwanted sequencing changes, whether certain regions are preferentially amplified, and then also the many potential effects of primer bias. And so you may have the option of moving to a PCR-free library kit, and an alternative is always introducing molecular barcodes, such as in Nugent's single primer enrichment technology. And importantly, if you want to detect some of those more difficult and rare mutations, such as larger insertions and deletions, copy number variants, or translocations, you really want to evaluate whether this is feasible with your library protocol and your overall pipeline. And so what we've done is we've combined each of these types of variants into our early access HD 753, which we call our structural multiplex DNA reference standard. And so there you can use a single sample with well-defined genomic complexity to evaluate that workflow. And importantly, this standard also allows you to evaluate the genomic context with regards to GC content, which, as we'll find out later, is important in terms of both the library protocol, but also the sequencing platform itself. And so to move on to the next stage in the NGS process, we have to also consider the bias associated with the platform itself. And once the sample has been prepared for sequencing, there are additional molecular steps that occur on the machine in the actual data generation step of the assay. Okay, so now we're going to dive into the 3 platforms. So you have 3 common platform manufacturers that are more commonly used in laboratories across the world. These include the Illumina series, which includes the MiSeq, the NextSeq, the HiSeq, the X10, and most recently the X5. And then the Ion Torrent Personal Genome Machine, which is also known as the PGM, and the Ion Proton. And then finally the PacBio platform. And so each of these has advantages in terms of read length, output, multiplexing, and overall versatility. But across the board, there are platform biases that may occur. And some of these include user errors, where the platform is overloaded, doesn't perform optimally, which will result in poor quality data. You also have machine failure and reagent issues that are ubiquitous across platforms. But there's also these platform-specific biases that occur due to the differences in chemistry that each one uses. And so when we look at Illumina's chemistry, this makes use of clonal amplification to generate clusters of short fragments, which is shown in the figure on the left. And then these are fluorescently detected using sequencing by synthesis. So as you've got your terminal base, as it's deprotected, then all 4 fluorescently labeled nucleotides are flowed over the flow cell along with the polymerase. And so following the single base incorporation, the flow cell is imaged to detect what base was introduced into each cluster. So you can see sort of a hypothetical image of this on the top right, and then an actual image from the machine on the bottom. And so then the process repeats itself for a certain number of cycles. This number of cycles determines the read length, and then the data from each cluster is grouped together, and this is considered one read. And so in this approach, there are a few places where errors can occur. First, you may have substitution errors that arise when an incorrect base is incorporated during that clonal amplification, which could then be propagated throughout the cluster. And then additionally, with Illumina sequencing, it's been found to have a sequence-specific error profile, which is thought to be a result of single-strand DNA folding or sequence-specific differences in enzyme preferences. And so for more information on this, I would recommend checking out the paper by Nakamura, which goes through this in a little bit more detail. And so when we consider ion torrent sequencing, we're again using sequencing by synthesis. And here fragments are prepared using emulsion PCR, again generating a clonal set of fragments, which are then deposited into a microwell on the sequencing chip. And rather than making use of labeled nucleotides and optics during the synthesis and detection, here individual nucleotides are flowed over the chip individually, again with the polymerase. And as the release of positively charged protons happen, these are electronically detected by a sensor in each of the wells. So the number of released hydrogen ions, and therefore the signal, is proportional to the number of nucleotides incorporated. And with this platform, if repeats of the same nucleotide, which are also known as homopolymers, are being sequenced, then you have multiple hydrogen ions released in a single cycle. And this results in an increased electronic signal, which can often be difficult to quantify for long repeats. And this limitation is also shared by other technologies that detect single nucleotide incorporations, such as in pyrosequencing on the Roche 454 platform. And so when you have this homopolymer bias, this results in insertion and deletion errors in the sequencing data. And so one of the newest technologies, the PacBio platform, this also uses a modified version of sequencing by synthesis. However, the difference here lies in that a single polymerase enzyme is fixed in each well, and so therefore single molecules are sequenced. And this is why PacBio sequencing is also called single molecule real-time or SMART sequencing. And so here the active polymerase is immobilized at the bottom of what's known as a zero-mode waveguide, or ZMW. And so then as fluorescently labeled nucleotides are diffused into the ZMW chamber, then you have illumination directed to the bottom of the ZMW, and the nucleotide that's held in that polymerase prior to incorporation emits its extended signal. And so that allows the base that's being incorporated to be identified. So when that nucleotide is incorporated by the polymerase, then the fluorophore is cleaved and it diffuses away. The specific base is based on the fluorescent tag that's detected, and so then the software generates a set of reads for each molecule. And PacBio Smart Sequencing touts impressive read lengths, averaging 10 to 15 kb in length. However, if the fluorescence signal is not detected due to quenching or non-labeled nucleotides, false insertions or deletions may be incorporated into those reads. And so, due to the nature of the single molecule detection of this technology, these errors are more prevalent than other technologies. When we compare to, for example, Illumina or Ion Torrent, where clusters or droplets of molecules are sequenced. And so this results in the error rate of the PacBio machine being inappropriate for the detection of low allelic frequencies, as they may be very difficult to disambiguate from the noise. And furthermore, the output of the PacBio machine is not yet at the same level as Ion Torrent and Illumina sequencers, but this does continue to improve. I will note, though, that the PacBio does show impressive resistance to GC bias. And so, as shown in the plots on the right, we see that the relative coverage GC bias for the MiSeq, the Ion Torrent, the PacBio in a couple of different genomes that have varying GC content. And so here, unbiased coverage would be represented by a horizontal line with a relative coverage equal to 1, which is shown here as the dashed black line. So PacBio does have a significant advantage in this area. And so while we see that the PacBio coverage levels are the least biased, and second to that would be Illumina, but all technologies exhibit error rate biases in these high and low GC regions, and especially in long homopolymer runs. And so, as I mentioned earlier, by combining data from two technologies, you can reduce the coverage bias if the bias in the component technologies are complementary and of a similar magnitude. And so in the data I've shown here, cross-platform replicates were compared. And so these DNA samples are from blood and saliva, and they've been sequenced on two different platforms, the Illumina and the Complete Genomics platform. And this resulted in an 88.1 concordance of SNPs across these replicates. In another study that considered Illumina, Roche 454, and solid sequencing, they observed 64.7 concordance. And so I think these two studies demonstrate the importance of considering the platform bias that you have in the pipeline that you've designed. So aside from cross-platform replicates, what other ways can we help? Can replicates help mitigate the error that's associated with our NGS assay? And biological replicates, which in this figure above are represented by an R, or samples that are independently prepared under the same conditions from the same host, as well as technical replicates, which are usually considered a repeat analysis of the exact same sample. So in the case of an NGS sequencing library, that would be your technical replicate. These both have significant utility during protocol optimization, protocol validation, and even later during your sample analysis. So biological and technical replicates can be used to assess your specificity and your sensitivity of your sequence variant calling methods, really in a manner that's independent of any algorithms and the chemistry that's used to call the variants, which helps you understand things like maybe your quality score thresholds or the parameters that you're choosing. So as I introduced earlier, technical replicates are usually considered a repeat analysis of the exact same sample. And so here, what we're using are the same sequencing libraries sequenced on the same machine using the same parameters. And so when you run these types of replicates during your initial pipeline development, or when you're evaluating new protocols or new sample types, you'll obtain information on the degree of noise associated with your pipeline. However, most laboratories would prefer not to use precious patient samples for these types of upfront experiments. And so by using Horizon's multiplex reference standards, this allows you to perform these experiments using a renewable, cost-effective resource. And so with these standards, you can determine the sensitivity, the specificity, and the limit of detection of your particular protocol, which helps you avoid false variant calls and also assists with data interpretation and your confidence that you have. And furthermore, as we mentioned in the Q&A session, Horizon reference standards are validated using a completely orthogonal technology, droplet digital PCR. And so that's really crucial for understanding the bias in your sequencing protocol. And so to wrap up the NGS workflow, I wanted to briefly mention again the role that bioinformatics analysis can play in introducing variability. And so I would recommend checking out under scientific support our archived webinars, where you'll find our bioinformatics webinar that was held last month. So that's where you'll find it. And so just to come back around to the figure that I showed at the very beginning of our discussion, if we make use of reference standards to better refine both the upfront molecular protocols and the downstream analytical pipelines, we can help improve the cost and the utility of next-gen sequencing. And so once we've accomplished that, what's left is really the downstream analysis. And as the authors note in this paper, linking a genetic variant to a phenotype, particularly a clinically relevant one, it requires a significant amount of expertise and effort. First, in identifying these highly competent variants, and then in estimating their functional impact. Finally, to be able to select among the functional ones, those that are correlated to the phenotype. And what you need there is an interdisciplinary team of bioinformaticians, statisticians, geneticists, biologists, physicians, all required to be on hand to translate the information and the primary data to useful knowledge and to understand the genomic variants in the impact of the genomic variants in the biological system. And this can take weeks and months of extensive experimental validations using things like downstream animal models or cell lines. And here at Horizon, we're a multidisciplinary team with experts spanning the range of translational genomics. And so at this point, I just wanted to hand things off to Jonathan to briefly introduce how our team fits into the Horizon offerings and the other resources that we have available.
Brilliant. Thank you, Natalie. That's a fantastic set of slides. I hope you all enjoyed that. So we'll sort of start wrapping up with I think it's four or five slides now, just to bring this all back around together. So at Horizon, we are a fully translational company. So we've got services and products that range all the way from genomics to translational genomics to personalized medicine. And so what we actually, the different range of areas we support and work with is in the basic research, drug discovery, drug development, drug manufacture, and molecular diagnostics. Typically, we've always focused with these webinars in the molecular diagnostic space. I should point out that all of our products are currently for research use only and should be used appropriately. However, because of the nature of this webinar and what's possible with next-generation sequencing, there may be actually also other interesting areas that we can support in, that you may want to dive in or ask us specific questions like cell lines for basic research or in vivo models, the generation of knockout mice, and all of those good stuff. And so it brings me around to the question I asked at the start of the webinar, and that is, what is the impact of assay failure in your laboratory, and how do you monitor for that? And in the molecular diagnostic or the molecular assay side of things, there are sort of three questions that we get commonly asked about, and these are questions that we are very passionate about supporting and very good at helping laboratories to answer. And that's, what extraction and quantification methods are you using? What is the true limit of detection of your workflow? And is the impact of formalin treatment interesting to you? What I've got here is a very nice summary slide of the 5 different groups of reference standards that we have developed at Horizon. And it's a bit of a teaser slide, so that if you want to know more about it, then please do get in touch with us. So on this graph, we've got on the y-axis the sample features. So that's, for us, that's how many mutations have we included in the sample. And on the x-axis, you've got sample complexity. And so that's going into either how well annotated that sample is, or have we put in, is there a lot of indels or insert or translocations or large chromosomal deletions, that sort of thing. And so where with the products we've talked a lot about is the quantitative multiplex reference standard, which is the circle the second from the left in there, labeled QMRS. That's got a good mix of complexity with the EGFR deletion. We've got some frame shifts, known frame shifts within the cell lines that are sometimes useful to testing the complexity of the next generation sequencing workflow. On the for people looking to dive into limit of detection and identifying how good a particular assay is, we have got gene-specific multiplexes, and they're available as DNA or FFPE. We've got our TrueQ DNA range, which has got a lot of features. So it's that range covers 40 different mutations, and there's 3 different tiers of that. So you've got a 5% tier, and you've got a 2.5% tier, and a 1% tier. We have, and we're very proud to be working within the Genome in a Bottle consortium, running out of NIST. So we have released the Ashkenazim Trio and Asian Sun as Genome in a Bottle FFPE samples, and so we're currently the only provider of the FFPE samples for that, and so you can certainly start using the Genome in a Bottle samples and using all of the NGS data that's available in the field. And then finally, on the right-hand side, and this is a standard we've just brought out, is our structural standard. So that does have translocations and copy numbers, and it's much more complicated than our other standards have been. It really will test your NGS workflow in terms of complexity. Like I said, it's very hard to cover all of the details in one slide. So if any of these if any of what I've said is like, oh yes, I want to learn more, just come onto our website. There's a certain amount of information on our website, and if you still have questions, then just live chat us or contact us.